Hi, and welcome back to Breaking Bio. I'm Morgan Jackson, a PhD student at the University of Guelph in Ontario, Canada.
I am Tom Housley, and I'm a postdoc at the University of Exeter's Penryn campus in the UK.
That is now the longest introduction we've ever had on the show from Tom. And this is episode 82. We're back, finally. We had a bit of a hiatus there, partially because people have been moving around, which hopefully will update everybody with all of our normal guests' updates in a future episode soon, so you guys can find out what we've been up to, because things have been changing. But that's not this week. This week, we're hanging out with Dr. James Gilbert, who is a lecturer at the University of Hull.
Hi, yeah. Okay, so I'm James Gilbert. I'm astonishingly a new lecturer at the University of Hull in England. I was previously a Marie Curie Fellow on a split site between Australia. I spent 2 years in Australia at the University of Sydney, and then a year at the University of Sussex in the UK. And I work on basically insect behavioural ecology with a focus on reproductive ecology. So I like parental care and also the evolution of social behaviour. And I'm moving kind of more into how that's affected by nutrition right now. So the interface of nutritional ecology and behavioural ecology. And in terms of what the kinds of species I work on, I'm kind of a bit of a jack of all trades, master of none, really. I've done a bit of work on lots of things depending on the questions. I did my PhD on assassin bugs in Uganda, which was quite cool. For the fellowship, I worked on socially plastic species. One was acacia thrips in Australia, and then there were some solitary or social bees in England. And it's I'm hoping to move more into kind of bee research now. But with this new position, I'm kind of trying to get lots of things in. So I've got some spiders coming in, and I'm going to continue work on thrips in the UK. I've got a collaborative line of work on bushcricket sex, which is quite interesting. I hope we'll have a talk about that in a bit. So basically, it's kind of a bit of a mishmash of lots and lots of things.
That is quite a lot of things to keep track of, and we'll leave the bushcricket sex for later. But you mentioned social plasticity, so I was wondering if you could just tell us what that is, what are the advantages of social plasticity, and what determines decisions on how social you are.
Okay, well, yeah, so social. Well, what the advantages of social plasticity are, I think, is what I'm trying to work out. We're talking about insects here where that are not your typical social, kind of complex social structures like a beehive. We're talking about insects where individuals make a decision: am I social or am I solitary? So they'll emerge from there, and they emerge as an adult, and they can either breed alone as a solitary individual, or they can breed in groups. And that breeding in groups can also mean several things. So if you're talking about a bee, you're talking about a decision made by offspring bees, whether or not to, in the case that I was looking at, whether or not to stay behind and help their mum reproduce, or whether to disperse and try to make a nest on their own. So if they stay behind and make their mum and help their mum reproduce, then you've got a typical eusocial kind of structure with a queen and some workers. But then in the other species I was looking at, in the thrips, you have a different set of decisions to make, where instead of offspring helping their parents, you've got what appears to be a situation where emerging offspring dispersing from a nest will make a decision: do I make a nest on my own, or do I collaborate with my sisters, probably my sisters, to establish a nest together? And so in these thrips, you have groups of 2 to up to 20 females will get together and make these nests together by tying leaves together with silk. And as far as we can see, it looks like you've got a set of co-founding females, and then they have a generation of offspring within that nest. What the benefits are is a wide open question. In some species of ants, there's benefits at the founding stage where there's a lot of competition between newly founded nests, and so collaboration makes sense in the light of conspecific competition. In other cases, individuals can make nests more quickly when they cooperate. In other cases, individuals can maybe kind of find prey more quickly or tackle bigger. In the case of spiders, for example, some spiders are social in order to enable them to tackle large prey items that they couldn't individually tackle, for example. What's happening in the Thrips case, we don't really know, and I think we're beginning to form a picture that they're maybe forced into cooperation by crowding. That's something that we call it. There's a hypothesis called the ecological constraints hypothesis, where the environment doesn't have enough breeding opportunities for individuals to go and all breed singly. So individuals are forced into collaborating. I was interested in testing for my fellowship an important alternative to that, which is the idea that it might not be poor environments that stop you from breeding individually, but simply poor individuals. Like if you personally are crap, then it might be the case that you're not good enough to breed on your own. So so you may be forced to help as the best of a bad job in a social group, and that's looking like quite a good hypothesis for these thrips. And that's where, this is where the role of nutrition comes in. As I said, I'm getting interested in the role of nutrition. I was interested in attempting to manipulate the nutritional environment for these individuals to see whether individuals that are given poor nutrition would then be more likely to become social. And it's a question that I'm going to basically, I'm going to keep hammering at that question until I manage to answer it. I didn't manage to conduct the manipulations that I actually wanted to do, either in the thrips or in the bees. So I'm basically going to keep going until I can find a system in which I can easily answer that question. That's what these spiders are about that I've got coming in.
That's cool, because I mean, thrips are, one of those orders of insects that practically nobody knows about, and you're probably one of a dozen, 2 dozen researchers working on globally right now. I mean, I don't think there are very many Thysanopterists around. Can you maybe explain a little bit for people at home that may not be familiar with thrips on what they are and how difficult I would see it being to try and change their feeding ecology and manipulating that, because from what I know they suck plant juices and stuff, which isn't a medium that would be easy to experimentally control in my experience.
Yeah, so this is so I have a byline in the talks that I give on thrips is adventures in non-model study species. And yeah, they illustrate quite well why we have model study species, because someone else has gone to the effort of finding the thing that's easy to work on. Yeah, so thrips are this delightfully weird order of insects that sit kind of in between the holometabula on one side, which have complete metamorphosis in an egg and a pupa in an adult, an egg and larva, a pupa in an adult, and then the hemimetabula on the other side, which have a series of nymphs, which each individually resemble an adult. And then in the middle you have thrips, which have this weirdo kind of development, which has what look like nymphs, but the last 2 stages are kind of quiescent. They're not quiescent. They're non-feeding, but still mobile stages that are called pupal stages, but which, to all intents and purposes, resemble a slightly weird nymph. And just to fit nicely with their weirdness, they're extremely tiny, and at that level of tininess, the air is like water. The air is viscous like water. So they have, instead of having wings, they have what are effectively oars, which is where wings would be. They have like a stick and kind of fringes of hairs on the stick, and they use that to kind of row through the air, and they've got fairly crap control of where they go with that, but they kind of like float around in a kind of a vague direction, like to all intents and purposes like a Daphnia in a water column. You know, they call them the aerial plankton because they get moved around with air currents, and that's a really nice way of thinking about it. They kind of have kind of a modicum of control over what direction they go in, but basically they're at the whim of the air currents. And the final wonderfully weird thing about them is that they have only one mandible. Their left-hand mandible, I think, is extended into a tube through which a stylet comes, and they suck the contents of cells through that, and they'll go down a line of cells like a corn on a cob, sucking the individual contents of each cell. They're just phenomenally weird.
Right.
So are all of them that tiny, and how does that make it incredibly difficult to work with them?
So they're not all tiny. If the ones I worked on were comparable in size to a Drosophila, I would say, but they can get up to. I also had around me, certainly in Sydney, the largest thrips in the world, which were about probably, 4 or 5 centimeters long, I would say.
Wow.
Which is a really weird thing to see, a thrips that size. And they're interesting in their own right. But the ones that I was looking at in the Outback were probably 1 or 2 millimeters long. That poses a few problems when it comes to I had to dissect them to assess their ovarian status, and that took a bit of getting used to. Because extremely tiny tweezers, and a lot of failure as well.
I always say that entomologists have got way more skills than neurosurgeons. They got nothing on us.
Yes, it was my first real kind of foray into the world of dissecting insects as well. So to start with thrips, I'm now going to be like, if I have to dissect anything again, I think I'm like spoilt for. Like, what do you mean it's difficult?
So you bring up that they're eating individual cells in plants. Brings me back to you trying to experimentally manipulate diets and stuff to try whether individuals are the ones that are giving up their breeding rights and just kind of going along with stuff and helping around the house.
Right.
How do you possibly manipulate individual cells?
Right. Okay. So I'll take you through the thought process.
Very quick.
And this is it actually forms the beginning of quite a nice story. I mean, I ended up with some nice research on these things at the end of it, but it was very different from the question I set out to ask. So the original idea was that if your thrips colony, if your thrips nest finds itself on a particularly crap plant, then if the plant is nutritionally crap as a whole plant, then your offspring will be more likely to cooperate when they disperse. That was the hypothesis. And so there were a number of ways I was going to attack that. One was to try and fertilize the plant, to see whether I could increase the lushness of the foliage, maybe increase foliar nitrogen, and that would maybe have a knock-on effect on the social behaviour of the colonies on the plant. So I basically might as well have tried to build a rocket to Mars, I think. That it was a perfectly valid idea based on my knowledge at the time when I submitted the proposal. But it turns out that the plant that these thrips live on is extremely, extremely slow growing. And So yes, this combination of factors makes this an utterly impossible dream. Extremely slow growing. It's a legume, so fertilization with nitrogen is likely to be totally absorbed by the activity of the rhizobia. And the outback is currently plagued with goats, which browse to the ground any young plants. So you have an extremely slow-growing plant. It's all over the place. Like, there's no problem finding individual plants, but they are all about 200 years old. So they're all fully grown. There's no chance of finding young ones to transplant and try and keep in the lab. There's no growing them from seed because they'll be kind of that big by the time the fellowship is finished. So basically the manipulation of the plant was not going to be an option. Another idea was that, and I thought this was quite cunning, the outback is full of creek beds, and it's also full of man-made roads, and in both of those places water, when it rains, forms these runnels, like either it's in the creek bed or water will hit the road and run off to the side. And it's a known phenomenon that acacia foliage on the side of roads is more lush than it is off the road. So that was one idea, was to go and look at thrips inhabiting the foliage of the specific acacia species next to roads and then off the road, to look at the effect of differential water availability. And that would be lovely if the sample size was high enough. But it turns out that these things, it took me absolutely months to find the very first colony I ever found of these things. So I bounced around for ages looking for viable populations, and I'd find, probably 1 or 2 colonies every 2 or 3 weeks. And eventually I found a nice big study population. But it was miles from a road, miles from a creek. So I was faced with this decision. Like, it was a bit of a marginal value theorem. It's like, is the cost of searching for a population which does encompass a road is the benefit of finding that population worth the cost of searching? And it basically wasn't, given the amount of time that I'd spent searching the population. Right, so so that left basically nothing I could do on the actual food plant. So I then tried to devise an artificial diet to feed them, which possibly may have worked. My supervisor there in Australia has this absolutely amazing protocol that he's developed. Is Stephen Simpson, whom you may have heard of. He's like this kind of granddaddy of nutritional ecology nowadays, because he's devised this system along with David Raubenheimer called nutritional geometry, which is an amazing way of asking animals to give us a behavioural answer to the question, what is your preferred diet? And so I wanted to eventually work towards using this nutritional geometry to to ask the question, if we imbalance the nutrients, does it mean you get more social? So I was trying to do it under that protocol and devising a liquid diet to feed the thrips through parafilm. And they appeared to be able to survive on parafilm, feeding on a Drosophila kind of based diet of varying compositions. But it turns out that they died too quickly from lack of humidity, to be able to make much inference about the diet they were feeding on. So
when you finally got the food figured out,
yeah,
it was the exterior conditions that screwed you up in the end.
And I realised that with about a month of fieldwork left on my first. And in fact, it was a really nice thing to do, because it ended up very nicely, because I was like, shit, it's the humidity, and that keyed me in to the idea that the function of the nest might be to maintain humidity. If you imagine it, you've got this kind of extremely flimsy silken nest on a tree in the arid outback, and it's housing these minuscule insects that normally, like by anyone's account, ought to be in a burrow or in tropical Australia. Except they're out on the tree, but enclosed in silk, and the tree is transpiring, so you'd imagine that within the nest there's a build-up of humidity. And with that month to go, I was like, right, I want to try and nail this, and I did an experiment to see whether it was humidity versus some other feeding style explanation for the nest. And I destroyed some nests, like by peeling away the silk, and I left other nests intact. And I put half of these treated, half of the treatment group went into a high humidity incubator, and half of them went into a low humidity incubator. And beautifully, the ones in the high humidity incubator, when I destroyed the nest, stayed alive for a lot longer than the ones in the low humidity incubator. But in intact nests, they just survived. Everybody survived the whole experiment. So it was really nice, and I got a paper out of that. But what it said to me was that those diet experiments may well work, but just only with humidity controlled.
That's cool. I like the fact that you set out to solve one question and found the answer to something totally different that is really, really, really interesting from a natural history and evolutionary perspective.
Right, exactly, and we'd previously not known very much at all about these insects. Bernie Crespi had come in before and done a lot of population censuses and gone, look, we have patterns of offspring production with different numbers of foundresses, and you get fewer offspring with more foundresses in this species, so there must be some kind of countervailing benefit of sociality, and here's the possible costs and benefits. And it was all very, very nice as a suggestion, but without much knowledge of the basic natural history of the species. And I think it's really nice that we're now able to kind of set a foundation with what the nest does, and we've got a couple of other papers about basic natural history of the species as well. From which we can now start to say, well, maybe this is the benefit, and maybe that's, for example, the fact that the nests need to be maintained is basically effectively parental care in this species, because the offspring can't make the silk of the nest. So the individuals in the nest are effectively maintaining it for their offspring, I'm a Dutchman if that's not costly behaviour. Like that's they obviously they have this behaviour, and you saw it at my seminar, Tom. They have this behaviour where they waggle their abdomen up and down in a kind of a twerking behaviour, which looks like kind of pumping the silk to their rear end. I think the silk is produced in their salivary glands, like that. From dissecting them, I'm not quite sure. But that's the hypothesis anyway. But I think they're pumping it to the right place, and then they have to lay these silk down in single strands, which collectively makes up a solid wall. And it absolutely has to be costly behaviour. And that for me is a very good candidate behaviour for helping as a possible function for social subordinates.
That's really cool. You'd think that even just being at the edge and doing this maintenance work, at the edge of the humid area, you're putting yourself at risk of drying out even temporarily. So, I mean, I guess, there's many costs to being the one on the edge doing the beaver work.
Right. Yes, exactly. So I found during the course of doing the fieldwork, I noticed there was variation in this in repair behaviour, and so I wanted to look at what determines that variation and whether it was linked to reproductive status. And that was why I had to dissect shitloads of thrips. And if there were going to be a typical social society, you'd imagine there'd be division of labour with some focusing on breeding and others focusing on helping. But in fact, the absolute opposite was true. Like, if you breed, then you contribute to repair behaviour. And of the ones that weren't breeding, some of them contributed to help, but a good proportion of them also, probably half the ones that weren't breeding, were not helping either, which is quite interesting. So the function of the ones that were helping, they're probably, I'm predicting that they were helping relatives, and we've got some genetics on the way to try and answer that question. But I was most intrigued by the lazy ones, the ones that were neither breeding nor helping. And one theory is that maybe they're waiting to breed. Maybe they are waiting to inherit the breeding position, upon which time they'll start reproducing and helping. And there's been a number of tests of that, and most of the time where people have tested it, they found that yes, things do start breeding when they inherited the nest. So I did a removal experiment where I found nests that were being constructed by two individuals, and I destroyed the nest a bit to see which ones were repairing and which ones weren't, and then I removed both of them and put one of them back, either the repairer or the non-repairer. And it turns out that if you replace the repairer, then they have loads of eggs, just as many as if the nest was built by one individual, and they also repair it very quickly. But if you replace the non-repairer, they take twice as long to repair the nest, which is putting their potential offspring in danger. And also, they don't really have any eggs either. So it appears as though they are simply crap. And so that's potentially it's not conclusive support, but it's certainly one kind of step in the direction towards the idea that crap individuals are forced to help as the best of a bad job, which is quite nice, I think.
That's very cool. It's more about thrips than I probably had ever known, and there's obviously tons of stuff going on there. But we should probably move on to giant testicles and male genital titilators for Tom's benefit. Because I'm sure Tom would be very upset if we didn't get to talk about some of these giant testicles.
Oh, absolutely.
Well, if anyone has a record-breaking testicular research, then talk about it.
Oh, this podcast got weird.
Yeah, well, I wish I'm going to have to disappoint you. That I hardly did any of the natural history work for those things, for those papers. I just did the phylogenetic statistics. But my co-author, he's the one that kind of unpacked these kind of stuffed testicles inside a tiny bush cricket. I think it was 14% of their body weight. Translated, we always love to hear the statistics for what it would be translated up to a human being. It would be, so that would be 2 human heads between your legs, I think.
Oh no! Oh, it's like the Elephant Man all over again.
How does it walk around? Well, they do quite a good packing job inside their body. So it's kind of rather than being dangling like ours, they're kind of packed in and around the body cavity. So they seem to have no problem jumping around.
So you do a lot of science communication and popular writing and on all manners of weird bug sex. Is that purely for personal enjoyment?
Absolutely. No, I totally love it. A lot of the time it's to push a particular paper that that we've got coming out. The one that I put one out that was to do with our most recent bush cricket paper, which was lovely actually, showing that what we previously thought of as a nuptial gift in these crickets, where in evolution it's been lost, in those species where it's been lost, the male has evolved these weird clasping devices that function to grasp the female and hold her down, and basically to try and prolong copulation as long as possible, which any sexual selection biologist would interpret that as a mechanism to protect his ejaculate, in order to try and stop the female from removing his spermatozoa before all the sperm has drained out into her. And that strongly suggests that that's what the original function of the nuptial gift was too. So instead of being something to do with paternal investment, to do with increasing the viability of the, to do with providing the female with nutrients, it's more to do with manipulation of the female and to delay her from removing the spermatophore. But that turned into, bush crickets like bondage. And if that generates a lot of readers, then I think that's absolutely fine by me.
You're giving me horrible flashbacks to a conference a couple of years ago where Kevin Judge was presenting some similar work on the sagebrush grigs, where the males have these gin traps that grab onto the females and then they try and push them off because the females eat the male hindwings.
There's wings in those species, doesn't she? Yeah.
Yeah. And so he got to the end of his talk and it was kind of coming close to time, and then he just flashed up this video and went, and now comes the ubiquitous cricket porn, and everyone was just
fantastic.
50 Shades of Bush Crickets.
Yeah, it's like that. It's all happening sexual selection wise. It's all happening in the Ensiphera, isn't it? Like it's crickets that have the mating frequency record as well, isn't it? It's something like 60 times a minute or something.
Sounds familiar. But it's also at the other end of the orthopteroids, the stick insects that are the long-term bonders as well for similar reasons.
Oh yeah. There's 70 days in one of them.
Yeah. Tons of fun stuff when it gets to orthoptera sex, it seems.
Yeah, absolutely.
So I think we're almost at a time, but I do have to bring it back to this. So one of the titles of one of your papers sounds all sciencey, and then there's a part in there that I think we need to explain a little further. So the paper I'm thinking about is increased copulation duration is associated with larger spermatophores and male genital titillators across bush cricket taxa. Can you perhaps go into a little bit of detail on what a male genital titillator is and how this works?
I wish we'd come up with that name, but we didn't. The titillators have been around in the literature for a very long time, actually, and they've been used in taxonomy for a while to distinguish between species, but no one has known shit about what they've done. So what we did was to what happens is the bush crickets have two sets of genital apparatus, these kind of non-genital structures which are around the male genitals. There's the cerci, which are the things which are modified into the gin traps and the handcuffs and the bondage apparatus. And then there's the titillators, which appear to have a slightly more romantic function. And while the cerci, the presence of these bear trap handcuff cerci, is associated with a long copulation after the transfer of the spermatophore, when the male is hanging on for dear life, trying to stop the female from shaking him off and getting rid of the spermatophore. The titillators, by contrast, are these little kind of projections, which can range from just a little kind of hillocks to really quite complicated toothed and feathery appendages. And the more complex the titillator is, the longer the copulation before the transfer of the spermatophore. And we hypothesized that that might be a period of mate assessment, where the male is either trying to stimulate the female to accept him, or is sizing up whether or not he's attracted to the female. And there's since been an absolutely beautiful piece of work on these things, on these titillators, by one of the co-authors on that manuscript, Gertrude Lehmann. She's managed to flash freeze a pair of bush crickets in copulation, and to see what these titillators are actually doing within the female. No one's ever been able to see that before. And it's a species with quite long and complex titillators. And she's got tomography results showing that the male titillator is actually titillating. Is actually doing that? It's kind of stroking some a particular internal part of the female, and then they went further and did histology on that part of the female, and they found that it's very, very dense in sensory cells. So it actually does appear as though the male is using the titillator to court the female during copulation. It's an amazing piece of work.
So they're ribbed for her pleasure, then?
Yeah, absolutely they are. Yes.
Morgan.
I may have just set this whole joke up. I don't know. That's really fascinating stuff. So you've got things working at all ends of the border, then.
Yeah.
That's cool. All right. Well, I don't know if we're going to get any worse jokes than that, so perhaps we'll just cut it off there. Where can people follow along with what you're doing at your new job, and all the things that you've been writing about insect sex and various other bits of parental care and thrips and natural history?
Well, okay, so you can follow me on Twitter. I'm James_Gilbert. I theoretically maintain an insect blog, which is Tiny Monsters, and that's at jdjgilbert.wordpress.com. But I've been very, very bad at doing that, and I've in the run-up to getting this job, which was a horrendous year of hell. I didn't update that very much, but I'm going to be better at that in future. In contrast, I've had quite a few articles on The Conversation, so you can follow me on The Conversation. The most recent one was about Father's Day, and how this Father's Day you should be grateful that your dad is a human being. And that's just about it, I think.
Awesome. Well, we'll make sure that we put links to all of the places you can find James, including some of his examples of his writing at The Conversation on our website, which is at breakingbio.com.
And I also say there's a TEDx talk as well. So if you could link through to that if that's a good idea.
Yeah, for sure. We'll definitely include a link to your TEDx talk, because who doesn't want to see more insect sex? So yeah, we'll definitely put all that stuff. We'll put it up at breakingbio.com, which is our home, where you can find all of our old episodes. You can also find us online at Breaking Bio on Twitter, and by searching for the Breaking Bio podcast on Facebook. And you can take us with you wherever you go by finding the Breaking Bio podcast on Stitcher and iTunes. And if you've got a couple of minutes and you want to go back and review some of our old episodes, we'd really like it if you could leave a rating and a review on iTunes. It helps more people learn about insect sex and the various other things that we talk about here. So do a favor for your neighbors and let them learn about this through outreach and Breaking Bio. So I want to thank Dr. James Gilbert again for joining us this week, and thanks to everybody listening at home for putting up with our spring hiatus, but hopefully we'll be back with many new episodes to come in the coming weeks, hopefully with less sex puns. No guarantees.
Never.